Hi, my name is Franck Barrat at the Hospital for Special Surgery in New York, and with my colleague and collaborator Jean-Charles Guéry from INSERM at the University of Toulouse, we'd like to introduce to you our recent manuscript that appears in March issue of GXMed that discuss how altered X chromosome inactivation at TLR7/8 locus can be involved in systemic sclerosis. So the work was really led by two very talented postdocs, Yong in my lab and Bérénice Faz-Lopez in Jean-Charles Guéry's lab. So systemic sclerosis is an autoimmune disease with a very high female predominance. It's a very complex disease that combines fibrosis, autoimmunity, and vasculopathy. The renal phenomenon is typically one of the first symptoms that patients can experience, but the skin thickening, but also sometimes involvement of internal tissues like the lung can have very, very large consequences for these patients. And as mentioned, it was very heavily described that systemic sclerosis is much more present in female patients than male, like about 80% of the patients are female. So we show in a previous study that plasmacytoid dendritic cells, pDCs, can infiltrate the skin of patients with systemic sclerosis. These cells, when depleted into bleomycin models, can prevent skin thickness and fibrosis. So there was a clear thinking that these cells can contribute to the fibrosis. And one of the ways they do this is by secreting this massive amount of type 1 interferon. And interestingly, pDCs produce type 1 interferon in response to toll-like receptors, and two of these toll-like receptors, TLR7 and TLR8, are actually expressed on the X chromosome. So the first thing that we did on that study is first look at the heterogeneity of the pDCs, and we use an enrichment method so we could purify enough of these very rare cell types and compared healthy donors and from systemic sclerosis pDCs. And as you can see here, there's a clear imbalance with the presence of subclusters number 0 and number 2, which are heavily represented in these patients. And when we did the analysis, we can find that these are really highly enriched in genes associated with type 1 interferon, which somehow made quite some sense. And interestingly, also, this cluster seems to be the more like mature and differentiated. So the premise that we had at that point, that's where we started to work with Jean-Charles Guéry, is to think about, well, what is the link between the presence of these highly enriched interferon clusters and potential dysregulation of the X chromosome associated TLR7 and TLR8.
So we previously reported that toll-like receptor 7 can escape X-chromosome inactivation in immune cells, and this is shown by RNA fish analysis, where you measure the primary transcript expression. We see here biallelic cells with two primary transcripts located on the active and inactive X chromosome that coexist with monoallelic cells in which you detect only primary transcript on one side on the active X, and the green signals is showed for this long non-coding RNA. So we applied this technology to pDC isolated for sclerodermic individuals, and we found that the frequency of biallelic cells for TLR7 was higher in sclerodermic pDC, as shown here by RNA fish. By combining RNA and DNA fish, as shown next slide, what we found is that TLR7 expression from the inactive X chromosome was associated with the looping of the TLR7/8 genomic region outside the XC nuclear territory, suggesting that reorganization of the chromatin could contribute to biallelic expression as compared to monoallelic cells. Next slide. So X-chromosome inactivation is initiated by the transcriptional repressor SPEN, which bind to the long non-coding RNA X to initiate X-chromosome inactivation, but it is also important for the maintenance of X-chromosome inactivation. And we measure XIST expression in a single-cell data set, and we found that XIST was downregulated in sclerodermic pDC, like SPEN. And interestingly, we found a negative correlation between SPEN and ESG score signature. So less you have SPEN, more you have ESG, suggesting that type 1 interferon may actually downregulate SPEN. So conclude, and as highlighted in the accompanying commentary by Girarca and Angera, our work provides evidence that impaired X-chromosome inactivation maintenance by deregulating and derepressing the X-linked TLR7 and 8 from the inactive X chromosome may contribute to aberrant type 1 interferon production by pDC in patients with scleroderma. So this may result in a chronic type 1 interferon signature gene expression. So as mentioned in this commentary, this may further destabilize the X chromosome inactivation machinery by reducing expression of SPAN or promoting the dispersal of XIST RNA beyond the XC territory. So this may act as a positive feedback loop of type 1 interferon signaling, which might be uniquely possible in individuals with two X chromosomes and could contribute to explain the female predominance of this disease. Thank you for your attention.
Yeah, and we thank you. I hope you enjoyed the manuscript, and obviously you can contact us if you have any questions.